Hi everyone. Today we're going to talk about DNA-based storage, and specifically we're gonna show you how we increase the density of DNA storage using composite DNA letters. So, storage of information has been a challenge from the very first day of computing, and in today's big data era, this is actually gaining very high attention since this is becoming a limiting factor. DNA is a very maybe good candidate to be the next generation of data storage. And in nature, DNA is used to store genetic information with very high physical density and robustness compared to what we know today, the state of the art magnetic tapes. Another advantage of DNA is the fact that it will never become obsolete. Unlike your old floppy drives or 8-track disks, in this DNA have been used in nature for the past 3 billion years and will remain relevant and readable for as long as we are around. So the basic idea of storing information on DNA is as follows. You start off with a binary file. Then you can encode it into a sequence of DNA letters, A, C's, G's, and T's. Now you need to somehow synthesize those DNA sequences into actual DNA molecules, which you can store in the fridge for as long as you want. And when time comes and you want to read it, all you need to do is choose your preferred DNA sequencing technology, obtain your sequences, and then decode the original information that you stored on this molecule. So how exactly does it work to synthesize DNA molecules with a desired sequence? So the basic idea is that you start off with a set of sequences, multiple sequences. Then you can contact any of the popular vendors there are in the market today. I will be demonstrating using Twist technology, but there are other vendors. They will synthesize the molecules for you and just ship it to your lab or to your home or whatever you want it. A tube containing your DNA sequences, molecules containing the sequences you want. So the technology of DNA synthesis works very much like what we know from inkjet printers. So you have a very small print head with 4 cartridges, one for each one of the DNA bases, A, C, G, and T. And in an iterative process, there is a linker that is connected to the surface. And then in an iterative process, in every cycle, the next DNA base is being injected onto the surface, and the molecules are being elongated as we go in each cycle. And when we finish the synthesis of the entire sequence we want, we can cleave the molecules from the surface and we get a tube containing the desired DNA molecules. So since this is an iterative process, there is an accumulating error that limits the number of cycles we can do. And the technology today is limited to somewhere between 200 and 300 bases or cycles in this case. As we saw, this is a very high throughput technology, so you can synthesize in parallel multiple different sequences, somewhere between 1,000 or 10,000 to 1 million different sequences that are being synthesized in one run of the machine. And as we saw in the cartoon, each one of the sequences that we are going to synthesize is going to be synthesized in multiple copies. And eventually we're gonna get one tube containing all those many molecules containing different sequences together. So to measure the density of DNA-based storage, we define 2 different measures. The first one would be the logical density, or the number of bits per letter of DNA sequence. This measure is also highly connected to the synthesis process, since in every cycle of the synthesis we synthesize 1 letter, and therefore is directly affecting the overall cost of DNA storage. and is limited by 2 bits per synthesis cycle, or 2 bits per letter, of course. The physical density would be defined as the number of bits per 1 gram of DNA. So in my tube, how much DNA do I have, and how many megabytes of information did I store in this tube? And today's technology allows us to get up to 500 petabytes per gram of DNA. Just to give you an idea of how dense this is, theoretically, we can store all the content of YouTube with just 1 teaspoon of DNA. So in a naive encoding, we take a binary message. Every pair of bits would be translated to 1 DNA letter. Then we'll have a DNA sequence that we can synthesize, and then later on sequence and decode to the original message. Just remember that those DNA molecules are going to have a very high sequence multiplicity. We're using or exploiting this high multiplicity in order to extend the DNA alphabet and increase the logical density, as we'll see in just the next slides. So imagine that we can take the 3rd letter of the sequence and design it such that it will contain a 1-to-1 ratio of A's and C's, meaning that if you look at the population of molecules representing this sequence, in the 3rd position, roughly half of them will contain an A, and the other half will contain a C. We can call this composite A's, a 1-to-1 ratio A and C letter M, and now we basically extended the DNA alphabet. So how do we achieve it? How do we actually synthesize this composite letter? We can do it by simply extending the printhead of the DNA printer by, in this example, adding 2 extra cartridges, one which contains a pre-mixed solution of G's and T's, let's call it K, and the other one which contains a pre-mixed solution of A's and C's, as we said, M. When we get to the 3rd letter in this fourth screen, when we get to the 3rd letter M, what we do is we inject this pre-mixed solution of A's and C's, and roughly half of the molecules will be elongated by A, and the other half will be elongated by a C. When we cleave it, we get a population of molecules that contains this 1-to-1 ratio of A's and C's in the 3rd position. So if we sequence a sample of these molecules, this 1-to-1 ratio will be remained. So we'll observe, in this case, 4 A's and 4 C's out of the 8 molecules we sequenced. So we'll be able to tell that this combination of A's and C's is actually an M. So the designed letter in this position was an M. If we look at the overview again, now we can take these 32 bits, let's say 33 bits. Since we have, in this case, 8 letters, 4 standard DNA letters, and 4 1-to-1 ratio composite letters, we can translate every 3 bits into 1 letter of our extended alphabet. And now 33 bits will become an 11-letter sequence. So the DNA sequences the DNA molecules that you have to synthesize in this case would be much shorter. And one other difference is that instead of just identifying the letter of each position, now we'll have to identify or infer the ratio of the 4 bases in each position. Okay, so let's put it formally. Formally, we can define the composite alphabet resolution. The composite alphabet with a resolution k parameter. In this case, phi k. Phi k will contain all the count vectors with 4 entries that sum up to k. The four entries, of course, represent the 4 DNA bases. The size of phi k would be k plus 3, choose 3. And for example, we can see phi 2, which contains the 4 standard DNA bases, together with the 6 1-to-1 ratio composite DNA letters. So if we want to infer a composite letter from the observed reads, in every position that we sequenced, let's say in sequencing depth n, we'll get a count vector that in this time sums up to n, and this count vector would be a random variable, which is a multinomial with a multinomial distribution, with n as the first parameter, and the probability vector would be defined from the original composite letter that we wanted to write. And what we want to come up with is a mapping function that will allow us to map every observed count vector to the correct composite letter from our alphabet. We tested two different mapping functions. The first one would be the LP norm mapping function, in which we simply takes the closest letter from the alphabet after converting both vectors to probability vectors or frequency vectors. Another mapping function that we tested was the Kullback-Leibler or the KL divergence mapping, in which case we took the letter that gave us the minimal KL divergence from the observed reads. This KL mapping or Kullback-Leibler mapping is actually equivalent to choosing the letter that maximizes the likelihood of the observed read. So if we derive the log likelihood function of the multinomial model and we derive the KL divergence, we see that the letter from the alphabet that maximizes the likelihood is the same letter that minimizes the KL divergence. This multinomial model also implies that there is a very strong effect to the sequencing depth. What we see here, or the sampling number, what we see here that if we sample with high enough sequencing depth, we can get up to 0 error in the mapping, in this small simulated example. So if we have errors, we have to somehow overcome errors. In this case, we define two different error measures, or two different error types. The first one would be sequencing errors or sequence errors, in which case we mislabel some of the letters or misread some of the letters. And to overcome those, we can somehow limit the legitimate sequences that we want to write, or we can use error correction codes to correct those errors. And the second error type would be dropouts, in which the complete sequence or complete oligo would be lost in the reading, and then we'll have to use error correction codes that are able to overcome erasures. In any case, any error correction code that we use will entail added redundancy and, of course, reduced density. So just to show you our result, what we did is we did a large-scale experiment in which we took a 6.5 megabyte file containing the bilingual version of the Bible. We encoded it into DNA, composite DNA of six-letter alphabet. We used Twist Biosciences synthesis to synthesize over 1 million oligos of the composite DNA. And we achieved a 25% increase in the logical density, meaning we actually saved 25% of the synthesis cost for this specific message, while we still had a physical density which is around 6 to 30 petabytes per gram, which is 6 orders of magnitude higher than what we know today. So the error correction code that we used, or the scheme of error correction we used, was the fountain code for dropouts and Reed-Solomon codes for the barcode and the payload parts of the composite DNA. And if we want to discuss the performance of our system, so we can see that if we look at all hundreds of millions of positions that contained an M, we see that the distribution of frequencies that we saw, or ratios that we saw, was shifted a little bit from the 1-to-1 ratio that we expect in this green line. And we can see that sometimes some of the positions contain only C's or only A's, despite having designed to contain a 1-to-1 ratio. And if we look at the KL boundary or decision boundary between the resolution 10 letters, 5-to-5 ratio to 4-to-6 ratio, we see that a significant amount of the positions would have been inferred in a resolution 10 experiment. We wanted to test the potential of the cost reduction that can be achieved using this method. And we performed a cost analysis in which we analyzed the synthesis cost and the sequences cost of composite alphabet of the composite system using different resolutions. You can see that as you increase the resolution, the alphabet increases, and then therefore the synthesis cost reduces. But on the other hand, in order to achieve a high enough inference rate, you have to increase the sampling or the sequencing cost. And the sweet spot or the trade-off is optimal in resolution K, which is a 50 alphabet composite resolution, achieving a 50% reduction in cost. This is, of course, using today's cost estimation. So to sum up, we saw that DNA storage can have very high physical density. We show that we have to use specific error correction codes for DNA storage. We described the composite DNA approach to extend the alphabet and increase the density, and discussed a little bit about the potential cost reductions. And by that, I want to thank my PhD advisor, Professor Zohar Yakhini, and the Yakhini group for very successful collaboration. The lab of Professor Roee Amit from the Technion, who did all the work for this project. Twist Bioscience for the collaboration on the synthesis of composite DNA. And the Adams Fellowship for supporting my PhD.